Okay, well, we're just about on about the hour, so I think we won't keep people anticipating this fantastic webinar any longer. What I'm going to do is introduce myself. My name's Rob Fisher. I'm the head of sales and marketing for the SPIE online resource optics.org, and I'm very excited to be welcoming Ethan and the rest of the team from QUEL Imaging and of course Blaze Bioscience as well, for putting on this fantastic webinar on accelerating clinical translation with optical phantoms with insights from Blaze Bioscience.
Thanks for organizing this. It's great to have optics.org and SPIE help us with this platform to share some of the work we've been with Blaze Bioscience. Before we really get started, I just wanted to talk about a little bit about what our mission is at QUEL Imaging. I think a lot of the people that are on this call probably know, but just to reiterate some of the things that we do, really focus on manufacturing phantoms, reference targets that support the whole life cycle of R&D, clinical translation, getting imaging technologies, optical sensing technologies into the clinic. And so what we're going to be talking about today is how we work with our customers to identify some of these challenges. And in this case, we provided some customized solutions. And we're working with a lot of customers in addition to making phantoms and custom manufactured components to provide our expertise. Alberto and I both did our PhD at the Dartmouth Thayer School of Engineering in how light interacts with tissue. So we have a lot of expertise there, manufacturing, software analysis, and so we'll be talking about that today with Blaze. And so really a lot of this comes from trying to support building the whole field itself. So Alberto and I really have a passion for trying to help standardize this field and build consensus. And that's a lot of what we're talking about is these sort of uncharted territories of how do you characterize an imaging system? And that's something that we think about all the time at QUEL. And we want to share some of the things the discussions we've been having with Blaze. So just to give, we did the brief introduction, but Stephen Daly, President and CEO of Blaze Bioscience, we've been working with Blaze for almost since we started the company. So about 2021, 2022, somewhere in there, working on with them in their endeavors to essentially make a BLZ-100 or tozuleristide, which is a targeted ICG. And they've also are in the unique position to be developing their own imaging device, which David Kittle has a lot of experience. And as the founder and the sole proprietor of DK Biophotonics, has done a lot of the work working with Blaze as well to develop the imaging system and the platform that they've been using and characterizing other systems. So he'll be talking about some of those specifics and the technical details that we get really excited about. And then Alberto is going to bring it home at the end, talking about some of the stuff that we're working on going into the future and where we think the field should be headed, and should be an exciting conversation. So happy to, if you have any questions throughout it, I'll be monitoring the questions feed. I'm going to kind of step back and let everyone else present. And then at the end, we'll have a discussion. So feel free to shoot questions through the portal. And if we don't get to them, like Rob said, we will follow up afterwards. So Stephen, you want to take it away?
Yeah, sounds great. Thanks so much, Ethan. Thanks, Rob. We're really excited to participate in this webinar, share some of our learnings. We have been working with QUEL Imaging for quite a long time, so we're happy to share our experiences and answer some questions. Just a brief overview of Blaze. I've just got a couple slides just to put some context into Blaze Bioscience, where we are and what we've been doing for many years. You can see here from this slide, we were founded to address, similar to many in the audience, I'm sure, address problems in cancer surgery. We really feel that fluorescence imaging guidance is a future tool and staple for many different cancer surgery applications. And specifically, Blaze was founded out of Fred Hutchinson Cancer Research Center here in Seattle. We're a Seattle-based biotech company. And it was founded by Dr. Jim Olson and Heather Franklin. Together, they got together and founded the company. And it took, you can see from this timeline, it takes quite a long time to take a concept from an imaging agent into the clinic. just from the chemistry perspective and getting safety data and getting your first in human trials. And so we were able to accomplish that about around the 2013 timeframe, and then moved into around 2015. Really, in those years, really trying to figure out what are great applications, clinical applications, and this ties back to in those days QUEL didn't exist. This was sort of going around trying to figure out what the best clinical target is for your agent, but using devices that you could just kind of grab and hope that they're working. And so you're sort of going around here looking for good fits for your chemistry and also good fits for your imaging, and that's what Blaze was doing throughout this time period from 2015 up through even 2018. With Blaze, we focused on pediatric brain cancer as our initial indication, and around that 2017-2018 time period is where we got introduced to David Kittle and his team down at Cedars-Sinai Hospital, is where David did some of his initial work, and we acquired some technology with that relationship, and started creating what we call the Canvas imaging system. And we focused this imaging system in brain cancer in particular. So we'll get a little bit into those details. But really, this is our first foray. This was the first foray of the drug side of the business interacting with the device side, and really trying to understand these two applications and how you put them together and how they can work as a system. I think we were learning that the drug performance does depend a bit on the device performance and how these two interact for different clinical indications. And then we continued on with pediatric brain cancer throughout 2020. Got our drug designations, orphan drug designations, and the like, fast track status and things. And then completed a phase 2/3 study in pediatric brain cancer towards the end of 2023. And since that time, we've been working with the FDA on further refinement of a pivotal of a confirmatory study to get that indication onto the market. So that's just a brief overview, but you can kind of see on the drug side, obviously it takes many years to go from concept all the way through clinical studies. And through that learning, it's inevitable that you will cross this question over drug performance and device performance in your specific clinical indication. And so with the help of QUEL, I think that's just speeding up being able to decipher what works and what doesn't work. Next slide, please, Rob. Okay, so tozuleristide, just a bit on our drug. It starts off with a peptide. It's a chlorotoxin derivative, and it's selective for cancer cells. Typically a lipid raft kind of mechanism of action for us. It does get inside the cancer cells, and it has fast clearance from blood. As Ethan mentioned, we are ICG based, and we like many others enjoy this aspect of the safety profile from ICG and many years of use in the clinic. And we do hope that, and part of the design of the molecule was being able to take advantage of all these ICG based devices that are out there on the market. And I think like many, this is where the interaction of drug and device starts coming into play. If you advance one more button there, Rob will get into this. So we're an IV push, easy administer prior to surgery, anywhere from 2 hours, 1 to 2 hours, 3 hours before surgery. We have experience where surgeries have been delayed, and the imaging agent actually works quite well even over 24, 36 hours prior to surgery administration. We really want to emphasize a real-time visualization because of that clinical environment of things moving and interference. And so that high contrast, that signal to noise that David will get into between normal tissue and fluorescent tissue becomes key. And also where the drug is selective in the biology versus where the device sensitivity plays into the equation. Next slide, Robert. Okay, so when we stepped our toe into the device development arena, we really wanted to sort of interact with our application. We are in brain cancer. We're dealing, the main tool for the neurosurgeon is a Zeiss or a Leica large surgical microscope, and so we wanted to design a system that interfaces with that type of platform, but still provide the fluorescence information. There is existing ICG-based capability in these surgical instruments and surgical microscopes. However, we found that due to blood-brain barrier and other aspects, we found that the concentration of our imaging agent was too low for the vascular ICG-based design of these tools that are currently built into the surgical microscopes. So we needed a more sensitive instrument. We needed the ability to detect lower concentrations of ICG and to properly differentiate that tissue. But we didn't want to, or in early clinical studies, we would wheel a surgical microscope out of the surgical field, wheel an instrument into the surgical field, and all this, these aspects of kind of interrupting the surgical flow. And commercially, we looked at that as a very large adoption barrier. And so we wanted to be as seamless as possible for the neurosurgeon. And this is the current iteration of what our Canvas imaging system looks like, with an imaging head that attaches to the bottom of the surgical microscope. That is still part of the drape system, so it's still it's within the surgical drape, and it does not interfere with the surgical oculars coming through. And it's tied together with an imaging cable, with an imaging station and computer processing unit that then plugs into surgical monitors and a touchscreen monitor on that imaging station that can sit near the surgical microscope. So this is just the form factor that we got into and felt like this is the design direction that we wanted to go with the Canvas imaging system. Next slide, Bob. So clinically, we have been in over 250 patients with our imaging agent. We've got a great safety profile. I'm not going to get into all the details here of some of our key efficacy findings, but I think what's key is that most of our experience is in brain cancer and in breast cancer, and then our first in human studies were in skin cancer for kind of some early dose finding studies. Next slide, Robert. So here's an example of what we see with our Canvas imaging system. David will get into here, but really I think the key product design objectives is sort of where I think you need to start and keep focused on whether or not an imaging system is working for the specific application. For us, obviously, you need to see, first and foremost, you need to see the fluorescence and be able to differentiate fluorescence versus normal tissue. That's key. That's pretty obvious, but there's a lot of details in that. I think we'll get into that. We don't want to see a significant performance drop in real-time video rates, and so we'll get into that. You can optimize the system for still imaging, back table imaging. You can optimize. There's great systems that have unbelievable imaging, but you can't put it into a surgical field. You may lose that great imaging once motion starts taking place, frame rates, all of that design aspect. We wanted to have full video rate, great performance. So where did and then for us, of course, the no interruption into the surgical workflow is a key commercial design objective for us. So so really when you start saying, how do you know if you can see the fluorescence and is it good enough? This is where we feel QUEL imaging really helps us and translate engineering data into clinical data. We use these targets in an R&D as R&D tools. All of the the 9-well, the resolution target, the flatness targets. They really help make design trade-off decisions early on, sensitivity, limits of detection, dynamic range, crosstalk, all these kinds of engineering decisions. In verification and validation, once you get to a certain point, you're writing specifications, and in the design development world, you need to write these specifications and test to them, validate and verify that the system is actually performing. these tools have been critical in that area for us. And then what we engage with QUEL for a custom target. You can see that down there on the bottom right. For really as a QC tool, we wanted to ensure there's some FDA a bit regulations or interpretation, how we interpreted regulations, into wanting to be able to see tozuleristide. So instead of saying, oh, we're just like ICG, we'll just make ICG targets, we said, well, why don't we just make a tozuleristide based target? That way there's no question as to whether or not you can, the imaging kit system can image tozuleristide on a back table perspective. So we engaged with QUEL, made our own custom target with tozuleristide embedded into the target. So we use that as a manufacturing QC check and also a clinical use system check prior to surgeries. And then, of course, device comparison tools. You throw everything when you're trying to figure out what devices work best. You throw all of these tools, at the situation and try to image and try to understand everything. So that's a high level of kind of where we've been using these kinds of information. And I think from there, I think we can hand it off to David. He'll get into more of the technical details and probably why most of you are here is to figure out what's going on.
Okay, David, I've handed over to you.
Okay, do we have visibility? Yes. Yeah, so as Stephen mentioned, we've used these phantoms for over 4 years now and a lot of different applications, but I want to highlight how we've used them and also how to properly use them for an application, which Stephen hinted on quite a bit, that it's one thing to have a device that takes good images, but will it take good images in the environment that you're interested in? A good example of this is cell phone cameras. Are they the best camera out there on the market? No, but there's 5 billion pictures a day taken with them. So clearly they're of use to somebody when there's 5 billion pictures taken every single day. So what this really amounts to is you have two extremes where which of these devices is better. It really depends on what you want to do with it. You can't say one's better than the other. And in terms of fluorescence imaging devices, this is what it would correlate to, where you have two sides. One is really well suited to intraoperative, handheld, surgical field, sterile, and then the other one is a fabulous lab instrument, back table, animal imager, has a whole host of uses, but they're really on a different spectrum that they are useful for. So what this comes down to is a problem statement. So when you want to take phantom data, you need a good problem statement which defines your fluorophore. So you need a good understanding of your fluorophore and device requirements, specifications, and these feed into a design of experiment that you're going to use while imaging your phantom. This immediately leads into what you're ultimately interested in, is characterizing your phantoms to characterize your device, and then device suitability. So is this device suitable for a particular indication? How does it compare from this device versus this device? Which one is best? If you're only looking at the images, you're not going to come to a conclusion that's useful, because really what it amounts to is we're interested in entirely different applications across the board. So you might have on one side an intraoperative system where you need high frame rate, you need minimal motion artifacts, you need video rate, very high immunity to noise. The OR can be a lot of competing systems like NeuroNav, which is in the near infrared. So if you're doing ICG imaging, it'll flash. Patient and user safety is high priority, sometimes even more priority than anything else. So you have to meet all of these conditions, a highly constrained environment. Whereas then you have back table imaging, which is a completely different side of it, where in this device, like the Pearl Trilogy, it determines its own conditions. It can be a dark environment. It can determine what kind of excitation you want to use, how bright it is. You can do long exposures. You can use anesthesia if you need to. So these are the two extremes. Often you might fall in between even, or you need a device that really does work for both applications. So as you feed these into a system design or into how you're going to actually use the system, you could think of it as a simulation world, where you're using these phantoms to simulate tissues, to simulate how the system behaves. And ideally you take the real data, but actually sometimes simulations tell us things that the real data can't. You can get underneath a system, you can get inside of it in an area that you wouldn't be able to see otherwise. But again, it's only meaningful if you set the proper boundary conditions, which in this previous slide really referred a lot to how the system is used. So what this comes out to being is you need to know what your use case is, what the lighting conditions are, because it's an imaging system. It's all about your light and the light you don't want. So often there's more light that you don't want than light that you do want. Fluorescence imaging is a worst case scenario of that, where not only do you have excitation light you have to get rid of, but you also have to deal with this other light, which could be still several times brighter. And as Stephen mentioned, our Canvas device worked with a surgical microscope. So that surgical microscope had a massively white light, massively bright light that was continuously on. We had no control over that. The surgeon had to have it in order to see during the surgery. So you have this bright white light, it's causing fluorescence over everything. Autofluorescence, anything that's going to fluoresce is going to fluoresce with that white light. So that has to be designed into what you're interested in seeing. So if you take a phantom data in a dark room, perfect environment, static images, you're not really going to get a full picture of if the system is suitable for your indication. So once you have all of those conditions set and you have a good idea, then there's more of the characterization of your device. And I was pacing around the lab one day debating about what requirements we need to write for a new device. And I ended up at QUEL's blog, and I would strongly encourage you to look at their blog and see what kind of things you can find there, because I've been doing this for a long time, and I read this, I said, this is exactly what I needed. It pointed to this AAPM. It hadn't actually been published yet, but the preprint was out there. And this is written by a highly regarded authors in terms of all of the characterization that could be done on a device. So table 4 here shows the different all the way from image sharpness, depth of field, signal uniformity, sensitivity, limit of detection. And it goes through each of these in detail, defining just what it means when you capture those. However, it doesn't tell you, because it can't, what your application is. So you have to tailor this around your application, capture the data around your application, and build up what that's really useful. So to look at just a couple examples of what this data is, depth of field and resolution. So QUEL has developed this resolution target, which has fluorescent backing and an Air Force target, commonly used, very recognizable. Handy for depth of field. So this would be an example of looking at group 0, element 6, and then scanning in the Z. So as you scan in the Z, you can see what your depth of field is for the device. We chose a fairly shallow depth of field for our device because we're already working under a surgical microscope, and we didn't need a lot of depth of field. So this would be how you would measure this very easily. Resolution, same thing. So you capture a single image of this. QUEL has developed a Python library so that you can input your image into that and directly get out the data. So that would be these data points here. So you can get your CTF and then figure out what your Raleigh criterion is. Another, and this is what I would say is one of the most essential, especially during manufacturing and verification, is uniformity. So this would be the entire system response of the device over the field of view. And this was something that I had to do on our first device with an ICG bath. So I'd mix up an ICG solution, pour it into a bowl, try to make it level, and then stick it on the table and take a couple of images. Quite difficult. And our current manufacturer just simply could not do that during manufacturing. It was not practical. So I reached out to Ethan and I said, hey, I really need this target. And they came back with this uniformity target. And when we were writing our verification documents, unfortunately, the software hadn't been designed yet in terms of analyzing it, but this is a two-sided target. So the top side is a grid, so you can directly measure the distortion and other parameters as well as flat field. But what we did is flipped it over and used the back, which was just a nice uniform fluorescent area. And with that, what you can get is a map of how much variation you have over the field. Our requirement was simply that we needed the 50% central area to be plus minus 30%. So we didn't have a tight constraint, but it had to be within that, and we didn't want hot spots. So if you have an area that's not illuminated by the excitation, for instance, alignment issue of the excitation, I had this a couple of times on instruments where it just simply didn't illuminate an area. So now you've got a whole area that could contrive a false negative. You really need to know that in the design of the instrument. All right, so again, there's a lot of different things that we can characterize here, but I want to focus a little bit on sensitivity since this is a big one. Sensitivity is, thanks to this new AAPM article, much more defined. Sometimes we think of sensitivity, well, can you see it or not? How bright is it? And how bright the fluorescence is certainly important, but it doesn't tell you a lot about the signal quality. So this 9-well target is essentially a fixed serial dilution, which is so convenient because now you have a serial dilution that you can put in your backpack, you can take with you on the plane, you can put in your back pocket, and you know it's not going to change. I've had some of these for years. And other than photo bleaching, which is always a concern, but photo bleaching you can monitor, so I'm careful when I take data that I put a beam block, especially with the white light, and then only expose the target for a short period of time when I'm imaging it. And I've not found photo bleaching to be a huge issue as long as you're monitoring it. But with this target, if you simply look at your data, what you would get is a series of images. So our device had 5 different sensitivity levels. So at the most sensitive level, we could see on this, it's called S5. So this is a long exposure. It was 2 frames per second. So very low frame rate. And then at high frame rate, we'd have what we'd call low sensitivity or short exposure. So what can you really see at these different exposure levels? When you look at this under a normal linear display, it's actually quite challenging to determine what your limit of detection is here, because there's just the dynamic range is huge here. You're going from 1 micromolar to 1 nanomolar. So orders of magnitude difference here. What can be very helpful is to take the log of the image. And if you look at QUEL's website, they often show the log of images. And that's simply just that. You would take the log of the image and then window it. So log, you might get a maximum value of 5 in your pixel intensity, then window that 5 up to 255. And then you get this image down here on the bottom. And especially if you use a color map, that can even be more helpful in visualizing these low intensities. So now in a subjective qualitative review of this image, we can see that at full sensitivity, S5, we're seeing down to a fairly low sensitivity. If we're interested though in 1 nanomolar, so that would be the center bottom well, it's simply not there. So there's just very little data in there. But we're still not seeing the whole thing. So what you can actually do is plot out an mean value over an ROI for each of those. So, and this would give you a plot of intensity versus concentration. You can additionally do a fit to this, and QUEL has a great white paper on doing this, where you can fit a line to this. Because it's in log-log, it would be a linear fit to this. And they can determine how your system is behaving versus concentration. As you approach the lower concentrations, you'll deviate a bit from that linearity. In our case, this is with the Zeiss white lamp on at 75%, so it's extremely bright. And there's some background. We do subtract that background out, but there's some errors in that. Additionally, there's some leakage from the laser, so you get a little bit of a ramp up. Another way to view this that I found to be very useful is to break apart each one of these wells. So you would form a image stack, and the image stack in Z would be concentration. So you would have a single image of 1 well, and then your next image would be a reduced concentration until you get to the control. So you break apart this image as I've done here, and then each one of those images in the image stack is normalized from 0 to 1. So every one of these would have the same intensity. What that gives you is an image like this. So now, essentially, with a perfect imaging system, every concentration would look identical. but that's in a real imaging system, you're going to get photon, you're going to reduce photon, you're going to get increased noise. So as you reduce the exposure time or you reduce the concentration, you're going to get roughly the same result, where you get increased noise in the system and your background, your signal noise is going to be dropping. So now again, in a subjective analysis of this image, you can get a good idea of where your limit of detection is in terms of what your system is capable of for a particular concentration. This is where we can bring in system comparisons. So now, if you want to visually compare two different systems, you can look at these normalized images of each concentration well and say at S5 sensitivity for our Canvas device, this is what we can see. Compare it to a Lycor Pearl, and you get this bottom image. So that would be this image. I think it just popped up now. The bottom image there would be the Lycor Pearl. So this would be a gold standard. It's a very good device. It's a very good image, but again, it's under ideal conditions versus what we have at the top is what we would get under more of a real world condition. I want to talk briefly about interpretation of phantom image data and some things to look out for. So we developed a custom phantom with QUEL a year or two ago that was to be used more for clinical, which is a binary go, no go. So before you do a surgery, you would stick this phantom in, and if you see something that looks useful, then you know that the system is running properly. We've actually found this phantom to be a handy thing, again, as a back pocket phantom, where if you have an opportunity to detect an imaging system, you can just take a simple image and get an idea of the viability of an imaging system. On the left-hand side, we have a really good image, low noise, high contrast, background is really dark. And then there's three scenarios here of images that we've seen that show problems. So one of them is a high background. So this image is being either from leakage of the excitation or leakage of the white light, whatever it is, it's not isolating. So the spectral channel has crosstalk in it, which is going to give you a false positive. Another issue would be high noise, which isn't going to show up if you're just looking at large tumors, but if you're looking at a really small area with high detail and high variation, the noise could be an issue. And then another one that we've seen quite a bit of is post-processing. So this would be a threshold. So the threshold was too high. And this is common for systems that use a high concentration of ICG, and they just threshold the lower values because they're not really useful. And actually might be advantageous to do that because you don't want to see everything. It's not clinically relevant. For tumor imaging, this might be a real issue because now we completely miss the right hand on this. We don't see it at all. So what this can tell you is where is that threshold and is it clinically relevant for you? Lastly, another thing we've really found useful is looking at your regular phantoms. So this is just a shaving of an ICG. So just take a knife, shave off a little piece of something that's fluorescent, and then start imaging it. So it's a really small object. It's a very small volume. It's not very thick, so its depth is small. So even if it's a high concentration, it's still a relatively low fluorescence. So this kind of bridges the gap between these perfect phantoms that have really nice boundaries to something that's a little more clinically relevant, that's got a small bound that's irregular and your eye doesn't really quite know where the real boundary is. So as you're looking through the sensitivity levels here, if you look at a low sensitivity, you really can't see the object at all. So on the upper left, you know, it's pretty much just you're at the level of quantization. But as you increase your sensitivity, it's much more defined. And just a word of caution here. If you're taking metrics, so I've talked a lot about subjective and qualitative image, and then we talked about linearity plots. That would be more of a quantitative approach. Imaging metrics are great. SNR metrics, SBR, or so you would have our tumor, the background ratio, or contrast. But you have to be careful to not violate the requirements with these. So in the case of a low sensitivity, there's a lot of pixels that are actually zero. So if your ROI encompasses an area that has zeros in it, you're not going to get a good value for these metrics that have a mean in the denominator. because you're dividing out by 0. So you're going to get a fake high signal to noise ratio that isn't actually meaningful. So what we see on this plot on the bottom is for WF-SBR, the low sensitivity actually performs the best. And that's not true, because you're not really measuring, you're violating the mean value. Is skewed because of that 0 values. Whereas SNR, appropriately, where you're taking a mean over the standard deviation of just the signal itself, appropriately shows that at higher sensitivities, you're getting a lot better signal quality. So another that Stephen hinted here is video. So it's great to just look at stills and still image quality, but if you need video rate, so in terms of a surgeon operating, you need to make sure that the system behaves properly for a video. So in this device, this was our preclinical device, which was really geared more towards stills and biopsy points. We focused on the still imagery. And what you get here as you increase the sensitivity is a nearly useless video. So if I pause this, what you see here is long motion artifacts, streaking, misregistration in time. So the viz was continually updated at video rate, but the near IR overlay did not keep up with the video of the color image. So you're going to have registration errors, you're going to have blurring. It makes it difficult to use. Again, depending on your application, this may not be an issue. But if this is important, then it's something that needs to be measured. Consequently, we've made a new device that has much better video rate quality. So if I pause this video, you can see the fluorescence image on the left has virtually no streaking. There's a little bit of on the subtraction because, again, this is designed for use with a microscope white light. So that white light is inducing fluorescence that is subtracted off. And as that moves, you're going to get a little bit of a subtraction artifact, but it's fairly minimal. So I want to highlight that always check the video quality. Don't just get stuck in the stills world if that is something that's relevant to your application. So in summing this up, really what this comes out to is you might have access to a single device. What you really want to be able to figure out is what your surgical image is going to look like. But say you don't actually know what that image, and you can't take it. You can't just go, you don't have, your device isn't at that situation, in that scenario. So you do have, say you have one device, what you want to do is build up a data set that completely fills your boundary conditions and your requirements. So you take phantoms, you take surgical data, you would take in vivo, ex vivo, chips, so you know your EEM, so you know all the how your fluorophore behaves. So you're building up this database, and then you can form a good forward model and decide what the system needs to behave like before you start designing it. And then while you're in the research and development phase, you can use these phantoms throughout the research and development phase to make sure you're on track. And then as you narrow down and start locking down your design changes, you continue to build up more of what this instrument is going to behave like. And eventually, hopefully you can start taking some clinical data and validate that the device really did behave the way you anticipated. But as you build up more and more of this information prior to the design of the instrument as early on as possible, you can get a much better look into the future using all of these phantoms and come to an appropriate conclusion. You know, so it really is much more of a process at the end of the day, where you're supposed to write up these requirements, but you may not have any idea what those requirements are. So a lot of this phantoms and this data will feed into your requirements. You iterate back and forth, you refine those requirements, and then you continue doing this until you have a deep dive into knowing exactly what you need. And then you design your instrument around that so that you're not designing a tractor when you really needed a sports car.
Good, Alberto.
Perfect.
Thanks everybody for coming to the webinar, and thanks Blaze for such a great set of presentations and comments and insights into the characterization world. So I'll be talking a little bit about the work that we do and that we're working on at QUEL Imaging. So we're a relatively small but very expertise team that works on phantoms. Really, we're in the business of supporting the development lifecycle of biomedical optics technologies. And so this means through the academic development, through contrast agent development, and then also imaging system development, including both open imaging and minimally invasive systems. This also includes the verification, validation, and manufacture stages, as well as the post in the clinical trials, regulatory, and post-approval quality control. So we really started out from the Center of Imaging Medicine, as Ethan has previously mentioned, and then we got a foothold of lab space in White River Junction. We've been kind of growing and making phantoms and building tools there ever since. We really got our official start from a phase 1 SBIR to work on these ICG reference targets back in November of 2020. We also managed to get a phase 1 NCI contract to work on flow and biomimicking anthropomorphic phantoms, so really getting into the more complex biomimicking shapes. We went on to do a phase 2 grant SBIR that finished up this past April that really allowed us to expand the reference targets, finalize those designs, and keep working through quality management systems, standardization of optical characterization, and then also work on trying to get better cross-system comparison enabled. And then we're currently working through a phase 2 NCI contract on the biomimicking targets, and this includes anthropomorphic targets, more advanced perfusion phantoms, and then also tools for accelerating FGS development, including digital twins for fluorescence-guided surgery imaging. So when we talk about these targets and tools, we've really focused the initial development on fluorescence imaging, and these includes these quantitative fluorescence targets for measuring concentration sensitivity, resolution, depth sensitivity, and a radiometric emitter target that also enables quite a bit of characterization of system. And then we've also done visualization and anthropomorphic phantoms. We've also expanded into application-specific optical properties that are beyond just fluorescence imaging. These includes the OptiBlocks product line, where we can tune the optical properties from the visible all the way to the short-wave infrared, and we have pretty stringent characterization techniques for that. We have dynamic fluorescence flow phantoms with channels that you can flow liquid through that really enable the early development of applications with custom fluorophores or custom dynamic imaging modalities. And then we also work on services and tools. This includes optical characterization, fluorescence, excitation, emission characterization as well. And then a lot of work has gone also into Monte Carlo simulations for fluorescence digital twins. When we talk about all this work, it's really contextualized through a lot of work that's happened in the industry, and then a lot of work that's been led by academics and people on the FDA and NIST. And so I was mentioned before by David, this APM Task Group Report 311 really served as a lot of content on what to aim for with regards to building these tools and phantoms for system characterization. Recently, there's also been draft guidance by the FDA, which got published in January with comments posted in April 2025. And so in the appendix portion of this, they talk about the optical imaging device considerations. They talk about some of the specific characterizations that should be addressed. And then they go on to mention that imaging device characterization can be accomplished in most cases by using phantoms for quality control assurance. So it's really about developing these phantoms and tools and this consensus on how to build these characterization systems that we spend a lot of our time thinking about and working on at QUEL Imaging. So realistically, for these fluorescence reference targets, really what we're looking at is these set of performance characterization criteria that were outlined by the AAPM report. And that's where the different reference targets that we've developed and expanded just from the start off from the ICG, also to 800 channel emission suitable for Cytolux and iodide under applications, as well as for the 700 channel, which is more suitable for SGM 101 and Sci 5.5 applications. We've recently worked on a uniformity and distortion target to finish wrapping up the characterization effort part of things. We've developed open source image analysis code, which can be accessed. And then we also have a lot of different resources, use guides, directions for use related to these targets as well. There are things that are outside of the scope that become more application specific that we also try to tackle with custom phantoms. And there's things beyond APM and the FDA, like image metadata. How do we actually cross-compare imaging acquisitions from one to the next that we're also trying to tackle and see how we can come to a consensus as a field. Really quickly through the uniformity target, it's able to get these fluorescence uniformity mappings. It can also use the data sets to get the distortion throughout the field of view and create these maps. And we have a preprint, and soon it'll be published out in the JBO Journal of Biomedical Optics. We're working through the first set of revisions on that. Really, from these standardized phantoms, we go into the application-specific phantoms, and this is where this custom target that we collaborated with Blaze on that allows for application-specific things, in this case, for system evaluation and ongoing monitoring during clinical trials. And so we can customize the different optical properties so that we can cater to the specific application. Beyond fluorescence, we also have application-specific optical properties in dynamic phantoms. This can come in different anthropomorphical shapes, different mu-A's and mu-S primes, and then different complexities of the flow targets as well. When we talk about complex phantoms, we're really trying to push the limit of what's doable by developing different manufacturing modalities and capabilities. So I'll showcase some of these more complex phantoms that we're currently working on to really showcase the ability to cater to application-specific applications. So here we can take a set from a human scan, develop a phantom from that, and then computationally generate, for example, vessels or flow channels. Then we can 3D print with water-soluble material, so that now we can have embedded flow channels that envelop this complex geometry. So this is recent development of the past few months that we've done in this case. Another set of complex phantom is development of the ability to do heterogeneous and multispectral. So in this case, we have a phantom that has about 0.75 millimeter blood inclusions at various depths with colon mucosa material. So you can see those channels with increasing depth from left to right. And then the optical property tuning we wanted to do over a span of the visible. So here from about 400 to 700, we were able to do multispectral tuning to match the shape for this particular application that required this. And similarly for the blood, we're able to do this multispectral tuning. So really going from homogeneous single tune phantoms into these heterogeneous multispectral tuning phantoms to cater for very specific application needs when it comes to the complexity of these phantoms. Similarly, we're working on clinical training phantoms. So at this point, we have to really care about the tactile and the mechanical properties, the ability for surgeons to be trained on this. And it's really thinking through on the ecosystem of what does it look like to translate this into the standard of care, which includes training of medical students. It includes training of new surgeons that will be taking up these new imaging modalities as part of their practice. So here, just a color image and then a fluorescence image part of this. As part of all this, we really have this academic core coming from the Center for Imaging Medicine, and that's where we really started with publications there on the initial designs for these ICG targets, and then also 3D printing methodologies to be able to get these more complex shapes and application-specific phantoms. And from there, we've gone on to do 3D printed tumor phantoms. We talk a lot about different metrics in this paper here. We did a collaboration with Brian Polk's group on an updated paper on the assessment of open field fluorescence-guided surgery systems. More recently, we've published this paper on toward fluorescence digital twins. We published the methods for that. We compared over a range of absorption and reduced scattering values. And the nice thing from here is we have expertise in tissue optical properties. We've had staff, Maina, particularly from our team, work a lot on the development of this code. And then we get to manufacture phantoms and do this validation of the fluorescence Monte Carlo to what the actual imaging that we're getting. More recently, we've published on archive for the QUEL analysis library. Similarly, we're about to publish, but the preprint is available for the RUD target. And then we're also working on some radiometric characterization methods, and that publication should be coming soon. So really, we're focused on this acceleration of clinical translation, and we can look at it as having these standard reference targets that try to tackle the different needs of characterization, facilitating this cross-system development and open source code analysis to really converge on common metrics and how to measure them from these characterization phantoms. We also have worked a lot on developing application-specific phantoms and the characterization and manufacturing capabilities to be able to scalably offer these for the different applications, as well as dynamic phantoms, and this includes the flow, but also these clinical training phantoms that we're developing with regards to having the different mechanical properties that can be used for surgical training. And really, a lot of this is thought leadership and expertise that we're trying to build on the field through the privilege of being able to work with almost everyone who's part of this biomedical optics ecosystem. And so we try to provide full lifecycle support, and that is both standard and custom phantoms for that consultation of different strategy system characterization, as well as custom R&D services, including simulation services as well. So with that said, that's the end of my presentation. I think we'll have Ethan take over for thank you and questions.
Thanks, Alberto. And sorry, just checking, make sure the system's working. But thanks everyone for presenting. I also want to thank our funding sources. And Alberto, I think your slide is just showing the other window there. So we were this project was funded by the National Cancer Institute. So there's the contract here where we were trying to build consensus in the field, and this is part of this user group sort of let's discuss the challenges. So it's been really great to work with Stephen and David to kind of get their feedback from using our phantoms. And so for initial questions, I think maybe this is mostly for Stephen, but maybe also David. Your teams have used your own phantoms. Like we've been working for many years, and I think many of our customers make their own phantoms as well. And so sometimes there's argument of, well, we make our own phantoms. Why do we need QUEL? Like what do you see as kind of the benefit for maybe when you would choose to use your own phantoms? Like there's something you need to do there. Like what is the driving force behind that decision of when to use your own versus kind of outsourcing and getting some of these other phantoms?
Yeah, I can start, and then David, maybe you can fill in. So from a high level perspective, obviously there could be. We started with that, and we still have sort of a capability, of making your own phantom, embedding, ICG and epoxies and things like that. I think that it's not you can start there, but with it's the consistency, your own methodologies. Are you being consistent? Because if you're really hanging your hat on this data, you want to make sure that the variability between phantom to phantom is sort of not questioned. So that was one of the main drivers. And then for us, we really wanted our own imaging agent specific target in phantom, and we did not want to invest in rebuilding capability that QUEL had already set up. These are targets that, in all sense, for us are something that we wanted to outsource and just place orders and be able to receive, just from a logistics, aspect as well. But I'm sure David has some comments on that variability and different technical aspects.
Yeah, I think, Stephen, you hit on a lot of ones. Consistency, repeatability, how stable is it over time when you're not using it, and what this expense and time of doing something like this. And once you get into manufacturing and verification, like Stephen said, you got to hang your hat on this thing. And that's a whole project in itself where you're verifying what you need to use for verification. And that's no simple matter. It's a lot easier to have something that's already past those requirements that you can use, that's published in the literature. Like Stephen said, we've made a lot of phantoms, and they're fine for research and development, perhaps, where you just need to see something. Sure, I'll stick an ICG cube made from some properties or even an alcohol. I've done a lot just dissolving it in alcohol, and they work for a long time. Photo bleaching is fine, but I have no idea what it is. I don't have the pipette skills necessary to make a really good serial dilution. And there again, a serial dilution is only going to last so long. If it's in water, ICG, it's a few hours. So the value that we really find is not just all of that, but in addition, you need to help figure out how to use the phantom, which QUEL is really good at that as well. Where it's one thing to take the data, it's another thing to know how to actually use the data. And it's software evaluation is, I've spent a lot of time making MATLAB scripts for evaluating these phantoms, and here they've got a whole API that you can use, and that's a lot of man hours in developing that. So it's really, it's the support, it's the consistency, repeatability, and just having a phantom that is something you can really hang your hat on.
Yeah, thanks for all those comments. I guess another question is, Blaze made the strategic decision to work with an ICG molecule, and so there are a lot of clinical systems that are out there that can see ICG. What advice would you give to other groups maybe that are working? I know in your case you're looking at targeted fluorescence versus other indications for ICG. So even some of these clinically approved ICG imaging systems are not suitable for what you need for your indication. So what sort of advice could you give to other teams that are either trying to develop for their own specific indication or working with a whole different fluorophore?
The ability to see ICG and you know, our fluorophore is ICG based, so it's great, but it really comes down to that clinical application and the end use case, and you know, you know, how useful is that tool at the point of surgery, you know, and then those designs of those tools, whether it be, you know, endoscopic imaging or open surgery imaging and stuff, they all have been built with different trade-offs in their own, you know, for their own system, and a lot of them are based on vascular-based ICG, so which is large concentrations of ICG. So what we've seen is you know, large variability really between imaging systems and trade-offs that the product manufacturers have made for their own product. When you can, you know, so that led us to the whole canvas imaging system for neurosurgery. But as we expanded to new indications, you know, we look at the same thing that the rest of the industry, I think, looks at, which is, you know, can this existing instrument work for our imaging agent in that clinical application? And it's never straightforward. We don't know just based off of bench data. We evaluate. We do a lot of you know, some comparison data. We try to figure out, you know, how well is it suited and fit for purpose, and then. But you know, we get surprised when we go into you know, early clinical studies. For example, we've got you know, an early clinical study right now in head and neck cancer. We don't, you know, you don't know until you really start seeing it in clinic. So it's very difficult. I think it's probably the, you know, it's getting better, you know, with tools like you know, QUEL provides to help standardize and to see, but really, you know, if you're an imaging agent only company, you're at the mercy of the device manufacturers and what they try to optimize for, and that may not be exactly how your imaging agent is working in the clinic. So it's difficult.
Thanks for that insight, and I think we're at the top of the hour, so I think we're going to have to wrap it up there. If there are other questions or anyone has any other anything you want to reach out to us, we're available. We're going to be at a few upcoming events. I'm actually going to be in London, Rob, next week for that hot weather. Hopefully it'll cool down a little. And then we'll be in the fall. There are a few other events that we will be attending. We'll have a booth at SBI Photonics West. And there are a few other events that if you follow us on LinkedIn, there are a few things planned for the fall that aren't quite confirmed yet, but will likely be there. So we'll let all of our community know when we get that confirmed. But I want to thank again, Stephen, David from Blaze. Really great that you were willing to talk about your experiences, and Rob for helping us organize this event, and SBIE for helping with this platform. So definitely stay connected. And